Hi, I'm Andrew Nenka from the Department of Radiology at the Medical College of Wisconsin. Thank you for watching my digital poster for the 2021 International Society of Magnetic Resonance in Medicine virtual meeting. This work is titled A Multispectral Imaging Template of Hip Arthroplasty, showing the distribution of 3T versus 1.5T artifacts in the acetabulum of varying implants. Portions of this work were funded by GE Healthcare. Template spaces provide great utility for research in brain, prostate, hip, and other imaging research by enabling consistent cross-exam, cross-field, and cross-patient comparisons. Unfortunately, such a template does not exist for MRI of hip arthroplasty. The articulation of the hip provides significant challenges, and separate templates for the acetabular and femoral components can address those challenges. Herein, we present a template for the acetabular component of the instrumented hip because adverse tissue reactions tend to occur in the acetabular region. With this template, cross-field and cross-patient studies of imaging around hip arthroplasty can be facilitated. In a proof of concept, we hypothesize that there are increased artifacts at 3T versus 1.5T in metal-on-metal implants compared to metal-on-poly or ceramic implants. In this study, 10 patients with hip arthroplasty received clinical MRI exams with Maverick at 1.5T and were called back for a matched exam at 3T. This included 2 patients with metal-on-metal, 2 patients with ceramic, and 6 patients with metal-on-poly implants. The signal void from each implant was manually traced in the OHIF viewer, and the resulting point cloud from the RT struct file was rendered into a volume as the interior of the surface, defined by the radial basis functions fit to the point cloud. Registration was performed through a multi-step process. First, the center of mass of the implant signal voids were aligned through a simple 3D translation. Then a rigid body registration was performed. To ensure that the acetabular components were aligned, the registration procedure used a mutual information cost function evaluated in a region around only the acetabular component of the implant. This was achieved by slicing the signal void, region of interest, at its center of mass and preserving the proximal half of the region of interest. A dilated mask was then generated and the non-dilated mask subtracted from it. In this region of interest, the registration cost function was computed. A multi-resolution algorithm was employed for registration with the first pass including a 2-fold spatial downsampling and a 9.4 mm full width at half maximum smoothing. And the second pass included no downsampling and a 4.7 mm full width half max smoothing. This image shows registered 1.5T implant images of the patient's left, and it is clear that the acetabular region is well aligned while remote regions show significant divergence. With a defined template space, 1.5T and 3T images were aligned to the template. For each participant, differences across field strength of signal void associated with the implant were computed. The percentage of times across all participants that a voxel showed more artifact at 3T were shown as positive or red, and the percentage of times that a voxel showed more artifact at 1.5T were shown as negative or blue. In the top left image, this probability map is shown across all participants in the study. In the top right, this probability map is shown across all metal-on-poly implants. The bottom row shows a probability map for metal-on-metal and ceramic implants on the left and right respectively. It is clear that the metal-on-metal implants showed increased artifact at 3T compared to other implants, and ceramic implants showed minimal field strength dependence in their associated image artifacts. When the raw volumes of image artifact are considered and compared with a non-parametric Wilcoxon test, the qualitative results of the previous slide are supported. There is a statistically significant increase in image artifact volume at 3T compared to 1.5T in metal-on-metal implants. A trend for increased artifact with field strength is apparent on metal-on-poly implants, but it is not statistically significant. Finally, there is not a significant field strength dependence of image artifacts associated with ceramic hip replacements. In conclusion, we developed an algorithm for the registration of the acetabular component of hip arthroplasties in MR images. This allowed the generation of a template space in which generalized analysis can be performed. As a proof of concept, we showed field strength dependent changes in image artifact volume with differing implant constructions. Metal-on-metal implants showed increased artifact at 3T compared to 1.5T. Metal-on-poly implants showed a non-significant trend for increased artifact with field strength, and ceramic implants showed negligible difference across field strengths. This new template space can be used for further, more interesting studies of pathology in the vicinity of hip arthroplasty.